Vitamins and minerals, zinc. We will now discuss this mineral that serves an antioxidant role, particularly that we'll see as a component of superoxide dismutase. Here we see food sources of zinc. So we see that zinc is found in different animal products, red meat, seafood, primarily shellfish and poultry. Also, we see it's found in dairy and some plant-based foods, including whole grains and some vegetables. Here's a chart of the zinc content of selected foods. So we see based on this chart that oysters will have the highest content per serving. Also, we see that crab meat is another good source. Liver and chicken, especially dark meat, will also provide good sources, as well as beef, veal, and pork. So you can see a lot of animal-based foods are going to be containing higher concentrations of zinc. Here we will see the digestion process for zinc. So we'll see when zinc is consumed in food products that it may be bound to amino acids or nucleic acids, so that will need to be hydrolyzed from these compounds, and that will occur in the stomach and small intestine via the hydrochloric acid, as well as nucleases and proteases will be involved in cleaving the amino acid or nucleic acid groups. We see here the absorption process for zinc. So it's a carrier-mediated process, but there are several carriers involved in the absorption through the brush border membrane. Also, we see that with high intakes of zinc, it can be absorbed by passive diffusion or paracellular absorption. And we'll see here the diagram of the first digestion and then absorption of zinc. Also, we see the metabolism within the enterocyte and then the transport through the basolateral membrane into circulation. So we see that when consuming zinc, then it's primarily going to be bound to different amino acids or nucleic acids. So we mentioned how HCl proteases and nucleases will be involved in freeing zinc from those compounds. So with the activity of these enzymes, we have zinc being in the free form, which may either be taken to the brush border membrane for uptake through several carriers, or the zinc can be bound to different inhibitors. So we see primarily phytic acid, oxalic acid, polyphenols, etc. can bind to zinc, which would limit its bioavailability and promote its excretion in the feces. So next we see the different carriers involved in the absorption of zinc that would then be available for uptake through the brush border membrane. So we have first here this ZIP4 carrier standing for the ZRT and IRT-like protein. And there's a number of these ZIP carriers. We see here specifically it's the ZIP4 carrier involved in the absorption through the brush border membrane. And we also have other non-specific carriers, although it's thought that this would be the DMT1 carrier involved in the absorption through the brush border membrane as well. So we see the activity of the ZIP4 and potentially the DMT1 carrier bringing zinc into the enterocyte, which then may be involved in different processes in the enterocyte or be transported through the basolateral membrane into circulation. And we'll talk more about what may be occurring here in the enterocyte. We see here the secondary mode of absorption of zinc. So with high intakes of zinc, it will be absorbed through paracellular diffusion. So we can see this process of paracellular diffusion between the enterocytes. And we saw paracellular diffusion previously with calcium as well. So we will now review the fates of zinc once it has entered the enterocyte. So we can see there are going to be three main fates that we'll review. So it will either be used functionally within the cell, it may be stored, particularly in this protein metallothionein. So it can be stored in metallothionein. We also see that it is taken up by certain vesicles or in the trans-Golgi network. So we will review that in the diagram. or we can see that it is transported across the basolateral membrane through the ZNT1 transporter. So we can see that largely zinc, once in the enterocyte, may be used, stored, or transported out of the cell. We see that it is a complex process, and there are a number of reactions and processes occurring here. We're not going to go over each of these or all of the proteins involved, but we will see here that within the enterocyte, we see that zinc can be used for a function within the cell. It can be transported out of the cell through the ZNT1 transporter, so it can be transported out into circulation. We can see that it will be stored in metallothionein. We also see that it may be taken up by vesicles or the trans-Golgi network. We'll now review the transport of zinc through circulation. So we see that it may be transported in the bloodstream bound to albumin. Also, we could see it may be bound to transferrin, alpha-2 macroglobulin, or immunoglobulin G. So these are going to be some primary proteins that it may be bound to in circulation. So it will be a number of potential proteins that it may be bound to as it travels through the blood. We will review the cellular uptake of zinc in different tissues in the body. So there are a number of these zip carriers. So we saw the first zip carrier involved in the absorption of zinc. So that was the zip4 carrier that was embedded within the brush border membrane of the enterocyte. We see a number of additional carriers, 1 through 8 or 14, involved in the tissue uptake of zinc. These will be found in particular tissues, including the liver, the kidney, the muscle, the skin, and bones. Within these cells, zinc may be stored as zinc-containing metalloproteins. So we'll see the primary function of zinc, or a key function of zinc, is its involvement in this enzyme called superoxide dismutase. So with the superoxide dismutase, it's actually copper and zinc dependent. So the superoxide dismutase is called zinc copper superoxide dismutase. When we review manganese, we'll see that there is also a manganese superoxide dismutase. So the activity of this enzyme is that it has an antioxidant role. So it's important in our antioxidant defenses, specifically against superoxide radicals. So the superoxide dismutase is an antioxidant defense against specifically superoxide radicals, and we can see the oxygen radical here. And this is going to be having its activity particularly in the cytosol of cells, and also we'll see it in the blood and in the lymph. So primarily this is occurring in the cytosol, the blood, and the lymph. The manganese superoxide dismutase has its specific effect in the mitochondria. So that will be a key difference between the zinc copper superoxide dismutase and the manganese superoxide dismutase. We see here that we have the superoxide radicals, and we see with the activity of superoxide dismutase, then we have the formation of oxygen and hydrogen peroxide. Hydrogen peroxide additionally has a capacity to promote oxidative stress. So we'll see when we talk about selenium. Selenium is involved as well in an antioxidant enzyme called glutathione peroxidase. So glutathione peroxidase will interact with the hydrogen peroxide to further reduce its oxidative potential. We see other zinc-dependent enzymes, so carbonic anhydrase, alkaline phosphatase, carboxypeptidases, and aminopeptidases. So in addition to its involvement in superoxide dismutase, there are additional zinc-dependent enzymes. We see further an additional zinc-dependent enzymes here, and its role in various processes. For example, its involvement in matrix metalloproteinases. So its involvement in this enzyme, it actually assists with wound repair or wound healing. So we see here these zinc fingers bind to the response element in the promoter regions of genes to increase or inhibit transcription. So you can see this interaction here, it to binding to the response element and then affecting transcription, the process of transcription. And we have seen this previously with vitamin D. So we have the vitamin D response element, so it's interacting with the response element here, and the vitamin D receptor, thus playing a role in the transcription process. The zinc fingers are proteins with a shape like a finger, and this shape is due in part to the presence of a zinc atom. So this is going to be an important role of zinc in addition to its coenzyme role, its regulation of gene transcription. We see here additional roles of zinc. So we see that zinc will play a role in cell replication. It'll be involved in bone, particularly bone formation. It will affect the integrity of skin. It'll play a role in cell-mediated immunity, as well as carbohydrate metabolism. And there's ongoing research evaluating the role of zinc in these and other processes that go beyond its coenzyme role and involvement in the zinc copper superoxide dismutase, as well as other enzymes. Particularly, we see new evidence for the role of zinc in cell-mediated immunity. There is evidence also in the role of zinc in reducing the duration and severity of cold symptoms. So it is interesting, some of this new and emerging research as it relates to zinc. We see the RDA for zinc. So we see here for men, it's 11 milligrams per day. For women, 8 milligrams per day. In pregnancy and lactation, it's 11 milligrams and 12 milligrams per day. And the tolerable upper intake level for zinc is 40 milligrams per day.